And osmosis, in plant, the cell shape is more concentrated than water. Therefore, water moves inside the cell, and this inward movement of the solvent from the higher concentration, that is outside, to the lower concentration, that is inside of the solvent, through the plasma membrane is known as endosmosis. And exosmosis, the outer movement of the solvent molecule through the plasma membrane from the higher concentration of the solvent inside the cell to the lower concentration of the solvent in the outer side is called as exosmosis. A solution is homogeneous mixture of the solvent and solute. If the sugar is mixed in a water and it disappear, then it is called a solution. If there is a two sugar solution A and B, and if in a fixed volume solution A has more solute than in solution B, it is called concentrated solution compared to B, and solution B is called dilute solution in compared to A. On the contrary, the relative concentration of the solvent in a solution B is higher than in solution A. If follows therefore that in osmosis the solvent molecule diffuse from higher concentration, that is dilute solution, to the lower concentration, that is concentrated solution of solvent. And what will happen when we put the animal cell into the solution of a sugar? And one of the following three things could be happen. If the medium surrounded the cell has a dilute solution, it has the higher water concentration than in the cell will gain water by osmosis, and such solution is known as hypotonic solution. If the medium surrounding the cell has exactly same water concentration as the cell and there will be no movement of the water, such solution is known as isotonic solution. And the third one, if the medium surrounding the cell has a lower concentration of water than the cell and the cell will lose the water by osmosis, and such type of solution is known as hypertonic solution. And this theory is described by the activity. In this figure we have seen the stand. This is the thistle funnel, here the egg membrane and the beaker and the water. Osmosis with a potato and the egg, remove the cell of an egg and take out the egg membrane, and egg membrane is tied to the lower end of the capillary tube, forming the egg membrane osmoscope, and the 1/3 part of the capillary tube is filled with a sugar solution, and this capillary tube is fixed vertically erect on the stand, and osmosis, that means osmoscope, is dipped in the beaker containing water, and the level of sugar solution is capillary tube is marked first, and you will observe that there is a rise in the sugar solution in the tube after some time. An egg membrane is semi-permeable membrane, and it allows the movement of solvent molecule through it, and when two solution of a different concentrated, that means concentration, are separated by the semi-permeable membrane, solvent molecule diffuse through it, and this diffusion is called osmosis. And in this experiment the sugar solution is more concentrated than water, therefore the level of sugar solution rises because the water enter inside the osmoscope by osmosis. Next activity described by the potato. The stand, beaker and potato, which is vertically hollowed. There was the pin indicator. A cavity is made in the potato after peeling it, and lower end of the potato is cut so that it can be rest vertically. Sugar solution is poured in the cavity, and the level of the solution is marked by using a pin. Now it is placed in the beaker filled with the water. You will observe that the level of sugar solution rises. Potato is made of the living cell, and these cells possess a semi-permeable membrane, and it allows the movement of the solvent molecule through it. As you know that when the two solutions of a different concentration are separated by the semi-permeable membrane, then the solvent molecule diffuse through it, and it is in this experiment the sugar solution in the potato is more concentrated than the water in the solution in beaker. Therefore, the diffusion of the solvent molecules from the higher solvent concentration at the water in a beaker to the lower solvent concentration, that is water in sugar solution, is known as osmosis.